All right, so in this video, we're going to be looking at Mendelian genetics. It's a big subject, but we're going to just kind of look at it as a cursory look, and then we're going to have some sections on non-Mendelian genetics as well, and also looking at some statistical analysis called chi-square, doing those in later videos. So just a little bit about heredity. Heredity is the information that is passed from one generation to the next. It's why we look like our parents. I assume that the baby in the left is the dad on the right. I think that's what I'm looking at there. But anyway, you get the idea. DNA and RNA are the carriers for hereditary information, and those nucleic acids, particularly the DNA, is transferred from one generation to the next. We looked at that as we looked at the process of meiosis and the process of making gametes. And so now we're going to be looking at how those genes are transmitted and how particular traits are linked to those genes. This is a conserved process. And so what do we mean by that? That means that this process has been passed down from one generation to the next, but not just from like parents to offspring, but parents to offspring for thousands and thousands of generations. And so examples of conserved processes are like all of the core metabolic pathways that we've studied in this class, like cellular respiration and photosynthesis. These are conserved pathways. The fact that all organisms have nucleic acids and these nucleic acids contain genetic information, whether you're a bacteria or a giraffe, this is true. All organisms have ribosomes, and those ribosomes make proteins which are coded for on the DNA. All of these are conserved processes which link all life together. So before we get into Mendel's law, it's important to kind of talk about that. So let's talk about things like genes, traits, and alleles. A gene is a unit of the DNA. It's a section of DNA that codes for a particular trait. A trait is just a genetically determined characteristic like hair color, eye color, height, hand size, ear shape. You could just keep going on and on and on that. And an allele is a specific version of that gene. And so you've all probably had this at some point in your biological studies, whereas there are certain traits, let's say hair color, and then you get that trait from your parents, but you may get one version from mom and one version from dad. Mom may pass you the blonde allele, and dad may pass you the brown allele, and then you end up with brown hair. Well, it's because some of those alleles always express themselves, and some of them all don't express themselves unless they're by themselves. We have these alleles called dominant and recessive, and so dominant alleles always express themselves. Recessive only express themselves when there are no dominants around. So, like a homozygous genotype, and we'll talk about genotype phenotype in a second, but a homozygous would be like homo, same, zygous, zygote. And so this is where mom and dad both give you the same version of the allele. This could be dominant or recessive. Well, if mom and dad give you a different version of the allele, that is hetero, different, zygous. And so dominant, again, always expresses itself. The genotype is what we just said, like homozygous, dominant or recessive, or heterozygous. These are terms that we would use with the genotype. Phenotype, these are words like brown hair and blonde hair. So the phenotype of an organism is the physical expression of that allele or that gene, and the genotype is the actual combination of those inherited alleles. The fact that the dominant allele always expresses itself is called Mendel's law of dominance. And we're talking about Mendel. I haven't even really mentioned Mendel, but he was a botanist and a monk who studied this. And so it's been called Mendelian genetics, named after this man named Gregor Mendel. And so there's genotype and phenotype. Again, genotype has to do with actual alleles. Phenotype has to do the expression of those alleles, the physical trait that you actually see. And then Mendel's law of dominance. The dominant always expresses itself. Here's a great example of this. Red is dominant, yellow is recessive. You mix red and yellow together, you get red, not orange, because red, in this case, expresses itself because red is dominant. Well, there's another law. There's actually three. So that was the first one. This is the second one, the law of segregation. So let's go back to meiosis to make this make sense. So remember, in meiosis, we had homologous pairs, we had sister chromatids. And so each chromosome you have is one from mom, one from dad. And you're going to get, let's say you get a big T from mom and a little t from dad. And you have that pair, and then you make sex cells, and you're going to make duplicates of those, and then it's going to separate. Remember, meiosis 1 and meiosis 2, and eventually that big T and that little t, the one that mom and dad both gave you, are going to separate. That means that you have an equal chance of giving that big T away as you do that little t. So that little t could somehow coat and express itself later. My mom has blonde hair, my dad has brown hair, I have brown hair, but I have a child with blonde hair because I had a blonde hair allele to pass down. That recessive allele was quote unquote hidden, and I was still able to pass it down because it's separated from the dominant alleles that are currently expressing themselves. And so that is an important concept. Mendel's law of segregation says that alleles will separate or segregate from each other in when they are inherited. And Mendel's law of independent assortment says that different genes on different chromosomes, independently assort themselves from one another. We talked about this already with how meiosis creates genetic variation. So here you have two different chromosomes, and you have two different genes expressed. This big chromosome has A on it, the little chromosome has B, and this one has a different version of B, and this one has a different version of A. Well, big A is going to go here. Little a is going, or yeah, big A, little a is going to go here. Where's big B going to go? Well, big B could go with big A, or it could go with little a. And so this idea is that each chromosome sorts itself independently of the other chromosomes. All the big letters don't always stick together. And so this creates lots and lots of genetic variation and different combinations. Now, as we look at this, we'll talk about Punnett squares in a second, but a Punnett square is just a way of doing this probability easily. But there are certain rules of probability that can help you without having to set up long, drawn out crosses. And it's just these two ideas. If you're looking at two things that are mutually exclusive, like an or statement, if A or B could happen, you add. So if you're going to get heads or tails, what would be the it'd be 100%, right? Half and half, 1/2 plus 1/2 is equal to 1. Well, if you're going to get a tails and a tails, well, 1/2 on one penny, 1/2 on the other penny. What's the chance that you're going to get if you flip two pennies? What's the chance that you're going to get two tails? Well, it's 1/2 times 1/2, and that is 1/4. And so that's how you decide that, and that's just pennies. But it's the same thing if you were looking at alleles, big A and little a, big B, or little b, and so forth. The easy way to show this with genetic probabilities is with something called a Punnett square. And a Punnett square is just a diagram to help you set up these crosses, but you could do this using laws of probability as well, just by looking at just taking the possible causes and multiplying them together. I'll give you some examples of those in class, or you could make this giant cross like this and do this. There are dihybrid crosses like you see here. Dihybrid just means that there are two different traits looked at. In this case, you got seed color and seed shape that are being looked at, or it could just be a monohybrid cross where it'd be just color or just shape. Dihybrid crosses are a little harder, obviously, but in reality, they're all pretty easy as long as you use those laws of probability.